The brain literally washes itself, and it does so mainly during sleep. Inside the tissue, between cells, there's a network of microchannels formed by the extensions of astrocytes, the glial babysitter cells. Their membranes are permeated by aquaporin 4, a protein acting as a gate for water. Along these pathways, cerebrospinal fluid is drawn into the brain's depths along arteries, mixes with extracellular fluid, and carries away waste products, from molecular debris to proteins like beta amyloid, outward along veins and to the meninge's lymphatic collectors. This glymphatic flow most sharply activates during the deep, slow-wave sleep phase. Neurons seem to momentarily shrink. The extracellular space expands by about tens of percent, making it easier for the flow to flush the tissue. Pumps are provided by vessel pulsations, respiratory micromovements, and the cortex's own low-frequency oscillations, which appear as a rhythmic tide at night. With age, the efficiency of this cleanup declines, and chronic sleep deprivation is statistically linked to the accumulation of toxic proteins. Hence the interest in sleep hygiene not just as a household tip, but as a factor for long-term brain health. It's also important that the brain lacks a classic lymphatic system within the parenchyma. Its role is taken over by glia and meninges, where lymphatic vessels have been discovered. This isn't a poetic metaphor, but physical plumbing. From body position to breathing rhythms, everything affects flushing speed. At night, the central computer performs a cache cleanup, an invisible service that runs without a single surge of consciousness, but with a measurable effect on daytime thinking clarity. Second fact: memory is not a cassette on a shelf, but a file that is rewritten every time it is opened. As soon as an episode from the past surfaces in consciousness, the memory trace becomes temporarily plastic. Reconsolidation processes are activated, and in this short window, new details can be written into it or old emotional marks weakened. This is why witness testimonies are so vulnerable to suggestive phrasing, and therapeutic techniques working with traumatic memories try to evoke them, then safely repackage and refix them. Biologically, this is not magic, but a fine coordination of the hippocampus, amygdala, and cortex. Part of the synaptic contacts, as it were, remove the lock, stabilizing protein molecules of memory marks are newly synthesized, and the recording is fixed in a slightly modified form. Emotions are powerful editors. Neuromodulators like norepinephrine amplify the engraving of significant events, which is why adrenaline receptor blockers in experiments weakened the affective coloring of some memories. From here also arises the myth of photographic memory. The adult brain almost never has stable, unchanging snapshots. There is a stitching of drafts that each time pulls in the context, smells, intonations, expectations. Paradoxical is the outcome. Plasticity, which makes memory unreliable, simultaneously allows learning, rethinking experience, and escaping the prison of intrusive images. The price for flexibility is inevitable edits, sometimes imperceptible, but always real. Third fact: the brain lives with a micro delay. Tens of milliseconds are needed for light from a lamp to reach the retina, signals to travel the visual tract, and the cortex to build a coherent picture. But we don't feel the delay, because the brain doesn't wait, it predicts. It constantly sends drafts of the future from the upper levels of the cortex down, comparing expectations with the influx of sensory data and minimizing prediction error. This architecture is known as predictive coding. When we make jerky eye movements, saccades, several such jerks occur every second. The visual system temporarily dulls sensitivity, saccadic suppression. And, in parallel, a copy of the motor command, corollary discharge, warns the visual areas about the upcoming shift of the scene. The result is a stable picture instead of a frame with blurry stripes. In another trick, the brain stitches together events that fall within a short binding window of about 100 milliseconds. Sound and image begin to seem simultaneous, although they arrived at different times, which is why a magician, by snapping their fingers, easily shifts our perception of the moment. Illusions like flash lag show that the system doesn't just chase reality, but anticipates the trajectories of moving objects in advance. Otherwise, we would constantly miss when catching a ball. Even semantic expectations work like a navigator. The brain completes noisy phrases, draws the ends of words, and predicts the next note in a melody. This constant advance provides speed, but sometimes results in phantoms. We hear what didn't happen, see a stable world where sensors twitch, and take careful substitution for pure perception. The conclusion is prosaic and surprising. The present moment is a meticulously edited version of the recent past, adjusted to the prediction of the near future. The fourth fact is that half of the brain's hidden power isn't neurons, but rather their shadow of glial cells, which were long considered mere passive support. Current estimates suggest the human brain has about 86 billion neurons and a comparable number of glial cells, and these glia perform functions without which thought would fall apart. Astrocytes, with their delicate tendrils, envelop synapses, forming a tripartite contact: presynapse, postsynapse, and the astrocyte itself. Their calcium waves and the release of mediator molecules, from ATP to D-serine, regulate signal transmission, like an invisible sound engineer adjusting volume. When the network runs amok, astrocytes supply neurons with fuel. They process glucose into lactate and, during activity peaks, feed synapses with it. Lactate also acts as a marker that triggers plasticity cascades, thus cementing memory traces. Microglia, the immune special forces and sculptor of connections, eliminate excess synapses during development and learning, guided by molecular tags of complement, freeing up space for desired circuits. Oligodendrocytes, by creating myelin insulation, control not only speed but also synchronization. Active pathways receive additional winding, and impulses arrive just in time to form a choral phase, the basis for clear rhythms and precise timing, for instance in the auditory cortex and hippocampus. The myth of solo neurons collapses. Without glia, energy doesn't function, sensitivity isn't tuned, and wiring errors aren't corrected. Even our nightly brain service, from ion balance to extracellular space cleaning, is largely orchestrated by glia, which has transitioned from the background to become one of the main protagonists on the neurological stage. Fifth fact: the brain is the body's most extravagant ascetic. It weighs about 2% of body mass, but at rest consumes, by estimates, a fifth of all energy, and operates at the power of a desk lamp, about 20 watts. Yet, it saves every spark. The main expense is not mental effort, but maintaining uninterrupted electrical readiness. Pumps in membranes drive sodium and potassium ions to store the possibility of an impulse at the edge of tension. Billions of synapses maintain gradients, capture mediator molecules, and nullify noise. Activity for a purpose, when you solve a problem, is merely an addition to the already enormous background. Hence the phenomenon of the default mode network. Even lying down with closed eyes, the brain expends a colossal portion of energy on internal simulations, testing scenarios, predicting social scenes, rehearsing neural circuits. The metabolic kitchen is finely tuned. Neurons prefer glucose, but during activity spikes, astrocytes convert it into lactate and supply it as fast carbohydrates directly to synapses. During fasting, the brain partially switches to ketone bodies, and this saves but does not eliminate the need for glucose. Energy dictates architecture. Coding is often sparse. Only a small fraction of neurons are active simultaneously. Inhibitory interneurons keep the network in check so that unnecessary impulses don't burn fuel in vain. And myelin sheaths speed up signal delivery without additional cost for every millisecond. Even rhythms, alpha, theta, gamma, can be read like a meters graph. By phases, they open and close windows of excitability, setting priorities on who gets current right now. The paradoxical moral is simple and strict. We see, remember, and imagine not so much because we have a lot of energy, but because the brain has learned to spend it with the parsimony of a jeweler, allowing itself the luxury of thought without exceeding the battery limit. The sixth fact. The legend of the grandmother neuron mocked simplicity, as if there existed a single cell responsible only for the face of one's own grandmother. Reality turned out to be subtler and more interesting. In clinics where people with epilepsy have very fine electrodes temporarily implanted in the medial temporal lobe, some neurons exhibit surprising selectivity. They consistently fire at the image of a specific person or place, an actor, a familiar building, even a fictional character, and remain silent to dozens of other images. Moreover, the reaction of some cells is preserved when the angle, size, or style of the image changes, and even when the name is presented as text or spoken aloud. Such concept cells are not isolated icons, but nodes in a very sparse coding. One entity is represented by an ensemble of neurons, and one neuron can be part of several ensembles, interconnecting stories and contexts. It is precisely thanks to this sparseness that episodic memory can quickly link a new event with existing concepts. You meet an actor in an unusual place, and the actor and Café by the Park ensembles activate together, encoding a new episode. An important detail: such cells are found in structures where the stitching of episodes occurs, the hippocampus and adjacent areas. In primary sensory zones, there are almost no such meaningful responses. The myth crumbles in both directions. There is no single magic neuron per person or object, but there are high-level detectors whose activity is linked not to pixels, but to the concept, experience, or story. This explains both the phenomenon of sudden recognition from a vague cue and why, when remembering a friend, we involuntarily complete their voice, smell, and favorite joke. What lights up in memory is not a photograph, but a network where meaning is more important than form. Seventh fact. For a long time, it was believed that nerves do not regenerate and that the adult brain is like hardened concrete. However, in one of its important zones, the dentate gyrus of the hippocampus, it seems to quietly continue seeding new cells even after maturity. Their source is stem-like precursors in the subgranular zone. They divide, give rise to neuroblasts, which over weeks extend dendrites, learn to listen to theta and gamma rhythms, and establish axonal contacts with the CA3 field. Initially, such newcomers are extraordinarily plastic. Their excitation threshold is lower, and their learning windows are wider, as if the brain is holding an accelerated integration course for them. The functional meaning is associated with separating the similar. When two episodes are almost indistinguishable, parking yesterday and today, two coffee scents, it is these young neurons that help differentiate the traces, preventing them from merging. In animal experiments, running, an enriched environment, and even the pulsating rhythm of new impressions increase neurogenesis. Chronic stress and excess cortisol suppress it. Antidepressants in some regimens, conversely, promoted the growth and survival of newborn cells, which was linked to a delayed clinical effect. In humans, the picture is more complex and controversial. Post-mortem studies yield conflicting results, from neurogenesis fades with maturity to it persists even in old age, albeit modestly. Therefore, be cautious with numbers. There are estimates of hundreds of cells per day for the hippocampus, but these are orders of magnitude, not markings on a ruler. In the olfactory bulb, where vigorous turnover occurs in rodents, the phenomenon is much more restrained in humans. And yet, even where new neurons are scarce, the brain is not hopeless. Dendritic spines migrate, synapses redistribute weight, and myelin sheaths mature. The secret lies not in the miraculous growth of memory, but in quiet gardening. Tissues know how to update their tools so that the network does not lose the ability to distinguish yesterday from today. Fact number 8. When surgeons, in the name of treating severe epilepsy, sever the corpus callosum, the main bridge between hemispheres, a patient's world doesn't fall apart, but one striking quality surfaces. The left hemisphere, where the speech center resides in most right-handers, becomes a narrator that cannot abide gaps. In classic experiments, stimuli were shown separately, either to the right visual field, processed by the left hemisphere, or the left, right hemisphere. If the word smile flashes from the left, the mouth stays silent, the left hemisphere ceases to speak. But the right hand reaches out to pick the picture of a smile, and the left, controlled by the right hemisphere, confidently points to the correct object. Ask aloud, "Why did you pick it?" And the left hemisphere provides an instant, plausible, but fabricated reason. Just in a good mood. I liked the color. This interpreter stitches together partial data into a coherent story so as not to live in the gaps between streams. In everyday life, the bridge is intact, but the mechanism remains. We often explain our own decisions post hoc, unaware of the hidden work of automatic perception and motivation systems. Facts from adjacent experiments add spice. In tasks with hints beyond awareness, people correctly chose options, inventing decent explanations. And in studies of the moment of intention, brain activity sometimes preceded the report "I decided" by hundreds of milliseconds, an important but arguably interpreted finding. In reality, this is not an annulment of will or a split personality, but a demonstration of architecture. Many specialized modules solve their private tasks, and the speech system hangs on a coherent subtitle. The goal of this subtitle is not truth at all costs, but the continuity of self, so that, day and night, by gaze and gesture, by right and left hand, we remain a single character who has reasons, even if he sometimes makes them up. Ninth fact. Pain is not a direct telegram from injured tissue to consciousness, but a decision the brain makes by comparing incoming signals with expectations, context, and the cost of error. At the very input of the spinal cord, the gate control theory works. Inhibitory interneurons in the dorsal horns either clamp the flow of nociceptive impulses or let it pass more widely. From above, descending control from the brainstem presses on these gates. The gray matter around the aqueduct and the ventromedial part of the medulla oblongata activate endogenous opioid circuits. Hence the phenomenon of placebo analgesia. When a person expects relief, the same descending system is activated, and the actual pain subsides. Administration of naloxone, an opioid antagonist, partially removes this effect, a direct indication of endorphins. The flip side is nocebo. Anxious expectation enhances the pain response and makes the brain more scrupulous about threat signals. Even the way the medication is administered is important. An open injection, when the patient sees the needle and hears the doctor's words, is noticeably stronger than a hidden one with the same dose. The body map in the cortex is also not passive. After amputation, some people retain phantom pains. The limb schema is still alive and sends alarm signals. Mirror therapy, by deceiving visual expectations, often breaks the pathological loop. In less radical experiments, the rubber hand illusion quickly changes the sense of ownership. The threat of a rubber hand is met by the body with a skin galvanic response, and pain thresholds in the assigned hand can shift slightly. In practice, this means that attention, beliefs, social signals, and even the gaze at one's own body are full-fledged regulator handles. The brain protects us from excessive damage, but it is not always a perfect engineer. Sometimes the protective setting becomes too loud. And one of the mysteries is how strongly word, ritual, and context can readjust the volume of pain as materially as a pill. 10th fact. Neurotransmitters are not emotions in a bottle, but a system of global settings that switches the network's operating modes. Dopamine, contrary to the popular phrase about the pleasure hormone, in key pathways rather encodes reward prediction error. Unexpected success causes a short surge. Over time, the peak shifts to a predictive signal, and the absence of the expected prize is an activity failure. On this math, variable reward slot machines and many digital reward systems thrive. Unpredictable likes and rare drops reliably train the brain to await the next chance. Acetylcholine from the basal forebrain increases sensory input precision. When a task requires attention to detail or the context changes predictably, cholinergic tone enhances data influence and reduces the imposition of internal guesses. The visual world literally becomes sharper. Noradrenaline from the locus coeruleus avalancheously rebuilds networks in the face of surprises. Short surges broaden the search, helping to shift from a habitual strategy to exploring new options. Pupil diameter, incidentally, often tracks this readiness for change. Serotonin, according to several models, sets long horizons and sensitivity to the cost of errors. It influences patience, suppression of impulsive reactions, and the balance of punishment-reward, although specific roles depend on circuits and remain debated. Together, these systems don't turn on joy or turn off sadness, but reconfigure weights in calculation, what to consider significant, how much to trust sensors, whether to go for risk or routine. A simple example, learning a new bike trick, dopamine learns from the expected received difference, acetylcholine sharpens the feel of the handlebars and road, noradrenaline flares up at an unexpected bump, switching to exploration mode, and serotonin helps not to give up after a couple of failures. The secret is that mood chemistry is a language of priorities, not emotion labels.